Welcome back to the second episode of Genetic News Now. Today we will explore the genius of Nirenberg and Mattaei along with Nirenberg and Letter. Nirenberg and Mattaei were the first to determine that a specific RNA sequence coded for a specific amino acid sequence. They utilized a poly-U homopolymer, cell-free extract, and 20 amino acids in which one was radioactive. The experiment deciphered the first of the 64 triplet codons in the genetic code by using nucleic acid homopolymers to translate specific amino acids. They then realized that if the protein was radioactive, it must contain the radioactive amino acid. This then enabled them to deduce the codons for the radioactive amino acid. Their final result enabled them to deduce the UUU codon for phenylalanine. Nirenberg and Mattaei determined that synthetic RNA made up a chain of multiple units of uracil instructed a chain of amino acids to add phenylalanine. The uracil chain, or poly-U rather, served as a messenger directing protein synthesis. Ultimately, the experiment proved that messenger RNA transcribes genetic information from DNA, directing the assembly of amino acids into complex proteins. In a later experiment, Nirenberg and Letter developed the triplet binding assay. They utilized a trinucleotide, a ribosome, and 20 charged tRNAs, along again with a radioactive amino acid. With the particular technique, ribosomes are bound to a single codon of 3 nucleotides, and the complementary amino acid charged tRNA was bound as well. This procedure allowed binding of amino acids to occur as well as the filtration of unbound amino acids. They were then able to check if the filter paper contained any radioactive traces. Their conclusion was that the filter paper is radioactive if the trinucleotide is able to code for the radioactive amino acid. This then enabled Nirenberg and Letter to identify 61 codons and ultimately give birth to the coding dictionary. This experiment was run on the basis that ribosomes, when presented in vitro with an RNA sequence as short as 3 ribonucleotides, would bind to it and form a complex similar to what is found in a living cell. The triplet RNA sequence acts like a codon in mRNA, attracting a tRNA molecule containing a complementary sequence. This complementary sequence is also known as an anticodon.
Well, that's all for today, folks. Thank you for tuning in to Genetic News Now.